Great, so I'd really like to thank everybody for being here tonight. And I'd really like to thank Mark Cook, Lewin and the Convergence Science team for giving me the opportunity to present to you all today. It's a real honour and privilege to be able to present our work to a much broader audience than our field alone. Now, I'm a clinician scientist. I'm an obstetrician, whereby I see patients in clinics, run the labour ward and also deliver patients on the labour ward and in theatre. And I'm also a scientist, whereby I'm in the laboratory being exposed to novel ideas, novel devices and the latest technology. Now, I really love what I do, being able to be at the coalface and see the most complex, challenging questions that we have in our field of obstetrics, and also being exposed to the new technologies that can potentially help us answer our questions in order to bring more mothers and babies home safely. Now, I work at the Mercy Hospital for Women. We're co-located with the Austin. We deliver around 6,500 babies a year. We're a tertiary referral centre whereby we have a neonatal intensive care unit. And this means that we see some of the most complex medical problems and for both mothers and babies. We resuscitate babies from around 23 weeks gestation and from around 400 grams, which is about the size of a can of Coke. Whilst most births and deliveries are absolute joy and an excitement in the room as their little ones being delivered, unfortunately for many, if a complication arises, it can be absolutely devastating and the worst day of our patients' lives. And perhaps it is little lives like this that have really spurred me and inspired me to delve into the science and into the laboratory in order to be able to improve outcomes for our mothers and babies. And it was really patients like Penny that drove me towards science. So Penny was a 26-year-old primary school teacher, and she was admitted at 26 weeks gestation because she had preeclampsia and her baby was also very small. Now, preeclampsia is caused as a result of the placenta not invading the mum's blood vessels correctly, and this leads to a reduction of oxygen and nutrients to the baby, but it also leads to that placenta producing toxins, which can lead to multi-system organ injury and failure in our patients. Now, unfortunately, we don't have a treatment for preeclampsia. The only way we can stop the disease is by delivering the pregnancy. Now, for Penny, this situation was all too real, as just 5 years earlier, she had had preeclampsia at a similar, very early gestation requiring delivery. And whilst her baby girl survived, unfortunately she had many complications of prematurity, including cerebral palsy as well as chronic lung disease. And now Penny was faced with the same reality that there wasn't a treatment to allow her to continue her pregnancy. And she was really facing the prospect of delivering another baby very preterm with those same risks of cerebral palsy and lung disease. And it was really cases like this that inspired me to go into the laboratory to try and find treatments and diagnostics for the most complex obstetric conditions. And I have so much gratitude and thanks to give to my supervisors of my PhD, Professor Stephen Tong and Associate Professor Tue Cacciolino. Throughout my PhD, we looked at developing novel treatments for preeclampsia. And my supervisors took me on, somebody who had been completely medical with no basic science training, to undertake a PhD that was completely laboratory focused. And I've really not looked back. I've really, really enjoyed my time in the laboratory. And it really was a bit like a scientific playground whereby you could come up with ideas, test them in the laboratory, and look, most of the time you get negative results and things don't work, but sometimes you get those moments of elation where something actually works. And for us, we found 2 possible treatments for preeclampsia. We've now extended those into clinical trial, both within Australia and South Africa. And we're still continuing research in that field. But now as a senior lecturer, I'm now expanding my research portfolio. And perhaps the best way to do this is to both look at the complex questions and challenges in your field, as well as look at the research landscape to see what is being developed and where the holes might be. And for me, I've expanded my research portfolio by looking at possible ways to monitor babies to prevent stillbirth. And the third theme is to improve monitoring in labour to again reduce risk to babies and to mothers. So firstly, we'll delve into monitoring to prevent stillbirth. Now, stillbirth is one of the most devastating outcomes in obstetrics. It affects 6 Australian families every day, involving over 2,000 Australian families every year. Despite advances in neonatal care, whereby we are reducing the mortality of our babies, unfortunately, the stillbirth rate has remained unchanged over the last 20 years. And perhaps this reflects the monitoring and a need for improvements in monitoring. Now, a spotlight was really shone on stillbirth by Christina Keneally just last year. Christina herself suffered a stillbirth in the 90s, and she got up in Parliament and gave a harrowing speech of her experience with stillbirth to Parliament. She asked a question whereby we need to have a Senate inquiry to further investigate why our rates are not improving and we're not better detecting these babies and reducing the risk of stillbirth. This came with resounding agreement between both parties that there definitely needs to be a Senate inquiry. And this has been undertaken, and it became quite clear that we really need to focus our research on developing better diagnostics in order to reduce the rates of stillbirth. Now, perhaps the focus of research to reduce stillbirth has really been in detecting small babies, as we know that these babies are at increased risk of stillbirth. So this graph here demonstrates the risk of stillbirth in babies that are small. The y-axis shows the stillbirth risk, whilst the x-axis shows gestation in weeks. And as you can see, the stillbirth rate in appropriately aged babies is much less than that of babies that are small. And the stillbirth rate remains consistently about 3 to 4 times higher in babies that are small compared to their appropriately aged counterparts. It's also really important to see that at term, so from 37 weeks, there's a steep increase in the rates of stillbirth for both babies that are small as well as for appropriately sized babies. Now, perhaps the reason why these stillbirth rates haven't improved is because of our detection techniques have remained largely unchanged. So how we detect babies that are small in the clinic is we use a tape measure. We measure the size of the mother's uterus, and that should somewhat correlate with her gestation in weeks. If we find it to be small, then we will refer the patient for an ultrasound to check on the growth. Now, unfortunately, this is our current standard of care, and it only detects 20% of babies that are small. There's been a real focus on research to see whether or not regular ultrasounds might improve the detection rate of small babies. And while serial ultrasounds do improve that detection, it is still just over half, meaning that around 50% of babies that are small still are not detected using these techniques. So if we could improve the detection of small babies, then this might lead to a reduction in stillbirth. But if we look at the causes of stillbirth, fetal growth restriction still only accounts for around 7% of those babies, with the vast majority of stillbirths happening in appropriately grown babies. And perhaps we need to think about why this might be happening. And whilst fetal growth restriction is caused because the placenta, we think that it's caused because the placenta doesn't implant correctly into the mum's uterus, and there's a chronic reduction in the amount of oxygen and nutrients getting across into the placenta. Perhaps the other reasons for stillbirth are as a result of a more acute fetal distress event. So the way that we can monitor for fetal distress in pregnancy is to monitor the fetal heart rate. Now, current technology uses Doppler, so an ultrasound-based technology, to monitor the fetal heart rate. We get a pattern, and there are certain patterns that correlate with fetal distress to show us that the baby is distressed, and that might lead to stillbirth. But unfortunately, current technology requires ultrasound, a Doppler, which is a huge machine. The ultrasound waves must be over the top of the fetal heart, so it requires a very experienced person to place the monitor, and then that baby can move. So quite often it will move out of the position of the ultrasound, therefore that Doppler needs to be moved. So it can only really be done intermittently. And unfortunately, for babies at high risk of stillbirth, whilst the baby could be coping quite okay 1 minute, the next minute that baby could be distressed, and we could completely miss that episode with the current technology. So perhaps a device that could continuously monitor the baby's heartbeat might be able to help us reduce stillbirth. And this was really the question that Stephen and I had. But how were we going to be able to extract that baby's heartbeat? So in actual fact, we need to use a different type of modality. And perhaps the best thing to do would be to develop a continuous fetal ECG that could be a wearable device that's used at home for patients at high risk. And this is where we engaged our electrical engineer colleagues, Professor Polani and Emerson Keenan at University of Melbourne. They've got a strong interest in developing the continuous fetal ECG. But unfortunately, current technology isn't that accurate, and there's a lot of signal dropout. So the questions that we need to ask is, why are we not able to continuously pick up that fetal heart rate? And why does current technology lead to such high amounts of signal dropout, whereby using current monitors, we're only able to pick up the fetal heart rate around 40 to 50% of the time. So the questions that we've asked is, potentially there's maternal and fetal factors that are inhibiting us from obtaining an accurate continuous signal. These maternal factors could be things like maternal obesity, and fetal factors could be things like the gestational age of the baby, the position of the baby, the amount of liquor, so the fluid level around the baby. The next question is the question of electrical interference. So this would involve things like internal interference, so such as maternal skeletal muscles, uterine muscle, as well as external interference, such as the mobile phone. And then the other thing that could be interfering with our fetal heart rate signal is motion artifact. So current fetal ECG technology is generally quite a bulky device that has wires, that every time the mother moves, there can be motion artifact, whereby we're just not able to pick up the fetal heart rate signal due to artifact from the actual monitor itself. So we've now embarked on a clinical trial whereby we've recruited patients in pregnancy that are having normal pregnancies, and used both traditional algorithms as well as novel algorithms that we've designed in order to eliminate those factors that can cause interference with this fetal ECG. And we found that using our algorithms, we're able to pick up the fetal heart rate around 84% of the time. And this is some data from our clinical trials. So the true fetal heart rate is the black circles that can pretty much completely overlap the fetal heart rate we're able to detect using our algorithms that remove interference from our signal. When we use traditional algorithms, we're able to pick up the fetal heart around 40% of the time. We're now extending this into a much bigger trial whereby we hope to recruit around 100 women, And in this next cohort of patients, we'll recruit women that have different complications in their pregnancies to see whether or not that might impact on the fetal heart rate that we're extracting. Potentially, one reason why we're not able to pick up the fetal heart rate the whole time is the current materials that we're using and the motion artifact that might be present. So on reading this paper by Aram and Garabans, this group is developing very similar technology to what we are. However, they're developing technology in order to extract gut electrical activity in order to improve detection of GIT problems. So they're developing a flexible patch-like device. It's similar to a tattoo that they'll be able to, an electronic tattoo that will stick on the skin, and it will be able to pick up signal for a longer period of time. So we contacted Greg O'Grady and Armen Garabans, and we're currently designing, using their technology that they've developed in order to manipulate it for our current use. So we hope that we will be able to produce a wearable patch that the mum who's at high risk of stillbirth will be able to wear continuously in order to pick up the baby's heartbeat. We're hoping then that it will be wireless and it will be able to alert the mum if there are serious problems with the baby's heartbeat by mobile phone to then attend hospital. And this would be a huge advance for us in order to try and combat and reduce stillbirth. So now the next research theme that we're absolutely focused on, and I'm really excited to talk to you all about, is developing better ways to detect fetal distress in labour. So the day that you're born is one of the most dangerous days in your life. This graph really shows you that the day that you're born, so this here is the risk of you dying on the day that you're born. It also depicts the risk of you dying through each year of your life. And it shows that in order for you to achieve a day that's just as risky of death as the day that you're born, you'd need to get to an age of 93. And that's in the UK and Australia. Now, if you're born in the US, the mortality rate's up here. You'll never experience another day as risky as the day of birth. Now, perhaps this is the result of the uterine contractions of labour. So as the uterus contracts, it reduces the blood supply to the baby. Each contraction then leads to a reduction in the amount of oxygen supply to the baby. Whilst most babies do have a good amount of reserve in labour, some babies don't. And this can then lead to that baby becoming starved of oxygen. That baby then switches from aerobic to anaerobic metabolism. And that then causes the baby to produce a high amount of acid. When that acid is produced, it very much correlates with poor obstetric outcome. And that can lead to cerebral palsy and stillbirth in babies. So the way that we currently monitor babies is we monitor the fetal heart rate throughout the labour. Whilst this is a continuous measure, unfortunately it's quite an indirect measure of how the baby's coping with labour. It takes a lot of skill in order to obtain the fetal heart rate, and it takes a lot of skill in being able to interpret and read the trace. Sometimes we can miss that critical point where the baby is hypoxic and could be delivered safely. And equally, sometimes the fetal heart rate might be abnormal and we'll go and deliver that baby. And in fact, that's the reason we deliver 30% of our babies by caesarean section. And the majority of the time, they'll be delivered. And once they're delivered, we realise that they're actually coping completely normally with labour as their acid levels are quite low. So perhaps a better device to detect fetal distress would be a device that detects a direct marker of fetal stress. And this device would detect the fetal acid level, as this level directly correlates with cerebral palsy and with poor neonatal outcome. So in order to develop this device, we contacted our physicist, Eric Shatner, and Georgina Sylvia, who's our chemical engineer, in order to develop a sensor that might be used during labour in order to detect direct markers of fetal distress. So they've developed a sensor that can very accurately detect acid levels. We've now shown that the sensor can absolutely detect acid levels within the physiological range, so that's from 7.4 to 6.8. We've also shown that the sensor is able to detect acid levels over a period of time of around 12 hours, as this is around the amount of time that labour can be. However, this work was all done in buffers. So next we need to see whether or not this sensor is able to detect acid levels in biological samples. So in order to do this, we've recruited many patients now and obtained the cord blood from the patients. We're then assessing whether or not our sensor can detect the acid level within that cord blood sample, and we're comparing it to a commercial blood gas analyzer to see whether or not the numbers marry up and whether or not the accuracy is similar. We're then spiking that blood with acid in order to drop that, to increase the amount of acid in that sample to see whether or not our sensor can also detect the acids in a comparable fashion to our blood gas analyzer. So this is one of our experiments whereby we've obtained the neonatal cord blood sample, we've added increasing doses of acid, and then we've assessed the level of fetal distress marker using a commercial blood gas analyzer. We then assess the levels using our sensor, and we've shown that they're almost identical. So next we need to place our sensor into hardware that we might then be able to attach to a baby during labour. Now in collaboration with Associate Professor Greg O'Grady and Dr. Robert Davidson in order to help develop the hardware for our sensor to be enclosed within, as our sensor is quite delicate. So next we need to see whether or not our sensor can detect fetal distress in an animal model. And look, we don't use animal models very often, and we absolutely only use them once we've got really good data to suggest that our sensor is working accurately. And we're in collaboration with Associate Professor Susie Miller. Now, Susie Miller is a fetal neurologist, and she's got a strong interest in cerebral palsy and reducing cerebral palsy in neonates. She's got a fabulous fetal sheep model whereby she performs a cesarean section on the sheep. She places catheters into the lamb in order to, so that she can test the fetal distress levels. She then obtains the umbilical cord of the fetal sheep and clamps that cord in order to induce fetal distress. And she can do this over a very short period of time to induce distress, or over a prolonged period of time. She's then delivering these animals, resuscitating them and using different treatments in order to reduce the impact of that fetal distress and hopefully reduce cerebral palsy rates in her sheep. So this is an absolutely fabulous model to piggyback on, whereby Susie can continue her studies while we can assess the level of fetal distress within the sheep. So Susie initially will induce mild hypoxia, and then by just clamping the cord a little bit and then releasing the cord, and then induce quite severe hypoxia over a period of around 8 minutes. Or she's got another model whereby she induces a small amount of hypoxia over a prolonged period of time. And we've currently assessed our sensor within these sheep a number of times and have really, really positive results, although we need many more sheep samples before we can be sure. So we're really focused on developing a sensor to better detect direct markers of fetal distress in labour in order to be able to detect those babies at risk of cerebral palsy or dying in labour, and equally to be able to save those women from an unnecessary caesarean section if their baby's actually coping quite well with labour. So we're now at a point whereby we are showcasing our science across Australia and New Zealand. We've presented in the US as well as around Europe, predominantly our preeclampsia work. However, we'll absolutely continue with our novel devices once we're up to that stage. Science has also allowed us to meet some of the real heavyweights in our field. So Gianpietro Palmero was at the forefront of developing the technology that we use in IVF. And Roberto Romero is in the US currently, and he is really at the forefront of developing therapies to prevent preterm birth. We're absolutely committed to trying to improve maternal and fetal outcomes through developing novel devices. So I thought that I'd leave you with a video from our research team.
When you try your best but you don't succeed, when you get what you want but not what you need, when you feel so tired but you can't sleep, stuck in reverse, and the tears come streaming down your face, when you lose something you can't replace, when you love someone but it goes to waste. Could it be worse? Lights will guide you home, and ignite your bones, and I will try to fix you. High above or down below, when you're too loved to let it go. But if you never try, you'll never know just what you know. Lights will guide you home, and ignite your bones, and I will try to fix you.
Great. So I hope that I've shown you that we're absolutely committed to improving the outcomes of mothers and babies. And I would really like to thank everyone at Mercy Perinatal, Mercy Hospital for Women, in particular Professor Stephen Tong, who's here today, Professor Sue Walker, 2A, Nat, Rox, Laura, Alex, who's here. And I'd also really like to thank our funding bodies. So we were lucky enough to get a project grant for the fetal distress in labour, or developing the fetal distress in labour device. Norman Baisha Foundation and Austin Medical Research and Cerebral Palsy Foundation. And I'd like to thank you all so very much for coming this evening.